Today we'll be looking at biodiversity. And what is biodiversity? Okay, it means it refers to, in an ecosystem, it refers to all the species within an ecosystem as well as all the variations within a species. So genetic diversity and many of the different ecosystems in an area. And there are good reasons to manage the biodiversity. We think of monocropping where there's just one type of species, and we think of nature reserves, or looking across from school at that mountain with the forest there. There's a diverse range of plants and animals there. But before we can manage the biodiversity, you have to be able to measure it. And ways of estimating the biodiversity in ecosystems. It's impossible to go out and count every individual organism and each species. And so you have to use different statistical methods, sampling methods, in order to get an estimate of the biodiversity. So you take a sample, and there are a number of assumptions you make, and you assume that your subset that you sample reflects the whole population, both in size and whatever the different species that you've looked at. Okay, so when you're trying to estimate the biodiversity in an ecosystem, it depends. The type of sampling that you do would depend on the type of ecosystem. And the organisms that you're measuring, are they sedentary? In other words, do they stay where they are, or do they move around? So sedentary things are things like plants and limpets and barnacles that actually stay put, and non-sedentary are things that swim around or walk around or fly around. Okay, so those things, if you're looking at non-sedentary organisms, then you'd be looking at some sort of traps. Okay, firstly looking at sedentary animals or sedentary plants and animals. And you would use if you're looking at plants, you use something called a quadrat. And these are pictures of quadrats. They're square frames of various sizes, but fixed sizes. So here you might have 20 centimeters by 20 centimeters, half a meter by half a meter, or a meter by a meter, and it's got a grid form on it. So it's a known area, and it can be placed over the part of the site to be sampled. And then you can count what plants or limpets or barnacles or whatever it is occur in that quadrat. And so here, sometimes it's impossible. Say if you've got a whole lot of different grass species and you can't count the individuals, then sometimes you use an estimate of cover. And so here, AC4, where A is abundant, C is common, F is frequent, O is occasional, and R is rare. So here, if we've got this area of grassland and we've placed a quadrat over it. And so the way to do it, you'd look, first off, this light green is everywhere, and that would be our most abundant species. And then if we're looking for the cover, say we look at the, here, for this yellow one, we want to see how many squares have got yellow in them. So you'd count all the squares with yellow in them, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, say 14 squares have yellow in them. And this is a 10 by 10 quadrat, and so 14 out of 100 squares have yellow in them, so you would say A has 14% cover. And sometimes there are different methods of dealing with these little leftover bits. Sometimes you look for ones that have kind of mostly covered, and then you'd have a lower estimate, or if you look for ones that have got a little bit in of it, then you'd get a higher estimate. But so you would do that for these two things, and then subtract it from 100% and assume that the background one has 100 minus these two bits in it. So you wouldn't have to count each one of the light green. Okay, where do you place this quadrant? Well, that'll depend on what sort of a survey you're wanting to do. Are you comparing two places and want to see what the difference between the two is? Then you would place the quadrant randomly in each square. And to find a random number, say here, this place here, say if we've got two quadrants here, this is 10 by 10, and this is like quadrant B, 10 by 10. And in order to find the square, you'd think of it being a Cartesian plane. Say this is 1 to 10 on the x-axis, and this is 1 to 10 on the y-axis. And you would have, say, two bags with little 1 to 10 pieces of paper in it. You'd pull one thing out of the x bag, and that would tell you your x coordinate. You would haul one number out your y bag, and that would give you a y coordinate. So in this case, this person might have hauled out 2 and then 3, and you would go to 2, 3 in that quadrant, in that sampling area, and that's where you'd place your quadrant. So this is your whole sampling area, and you have put a square over it, or you've put a grid over it, and you're deciding which areas to place your quadrant. So if this was not 1, 2 quadrants, this is your whole sampling area, and here in fact you've got 20 along here and 10 along this one. And so this would be 2, 3, and this one is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14. So if you pulled 14 out of your X bag, and you pulled 1, 2, 3, 4, 5, 6, 7 out of your Y bag, you would go to this little square and you would place your quadrant here and measure the grasses at this place. So that's using random sampling. If you're using random sampling in order to decide where to place your quadrant. Now, if you aren't looking just at comparing two places, two areas, then you would use systematic sampling rather than random sampling. And so for systematic sampling, you'd be looking at where you're expecting something to change in a gradient. So thinking about like at the beach, you might want to see, going away from the shore, how many barnacles there are on the rocks as you go further and further away from the sea. So you've got a gradient and you're expecting there to be more barnacles close to the sea and less barnacles further away. So then you create a line called a transect, and your transect line goes along the gradient that you're wanting to measure, and then you would take quadrants at regular intervals along the transect. So this isn't random like this one. This one you place a transect and then you take quadrants at fixed places along the transect line. Okay, so if you've got mobile organisms that you're looking for, if you've got fairly small animals, here you would use a pitfall trap. So if you say measuring frogs, you want to sample the frogs in an area, you might create a pit or a little, and what you do, you dig a hole, you put a glass jar inside it up to its rim, and then hope that a frog came along and fell down in here, and hopefully that the frog couldn't get out. Or perhaps a little mouse or something that wouldn't be able to climb easily out the jar. Or ants, perhaps you could get. So, but the thing one disadvantage of the system is it's not just measuring things that are there, things that are moving through. Depending how active that organism is, you might get more of those than you would get of less sedentary. If you get, the more active the organism is, the more of those you would collect. Okay, a pooter. This little pooter is for collecting insects, small little insects, and here, to avoid touching the insect, you have this pooter, you suck on this end, you've got a filter here so the insect doesn't end in your mouth, and this bit you put to where the insect is, and by sucking here you create a low pressure and it sucks the insect into the specimen jar and you can examine it in there. If you've got larger animals, you would need, thinking about larger animals, you might need a net, or you'd need some other sort of trap in order to examine larger animals. Or these days you can have a photo kind of a camera that is, what do you call those things? Photo traps. And those things that they look to see, how many leopards there are around, because nobody wants to sit there waiting for the leopard. You can have a photo trap as well. Okay, so looking at these methods, quadrats, transects, pitfall, traps. So advantages, okay, this is quick, inexpensive, and portable. Not always very accurate, and you have to take a large number of quadrats in order to be quite accurate, because say if you just did a few and happened to place them in places where there was no vegetation, if you were doing a grasses survey, you have to take enough of them so that to remove bias. Okay, transects are quick, inexpensive, and portable. Okay, but sometimes they're used inappropriately. So you've got to decide carefully whether it's appropriate to use a transect or not. If you've got two areas that you're just comparing to each other, you wouldn't use a transect. But say if you were looking at the grass species in shade going into sunlight, then that would be an ideal place for a transect. Okay, pitfall traps. Inexpensive, easy to set up and use, but quite often you kill the organism that you capture and you may oversample or undersample.